Okay, so I appear to be having a bad day today in terms of presenting what I wanted to present. So let's go back and fire up the pen. The word that I meant to have in here is microfilaments. And it makes more sense in terms of microfilaments. So rather than talking about microtubules, let's talk about microfilaments. What would you expect to happen if you block the function of microfilaments in frog embryos before the neural tube forms? So remember what the function of the microfilaments is to do is to anchor one end of the cell as the microtubules elongate the cell. Then once that elongation ends, the microfilaments contract to narrow that end of the cell and to fan out the other end of the cell. And that's what causes the cells to then roll out like this and roll to form the microtubule, the neural tube. So if you block the microfilaments from functioning, in other words, they don't contract, you'll get the elongation of the cells, but you're not going to get the rearrangement that narrows one end of the cell and fans out the other end of the cell. So these cells will not roll around to form the neural tube, and so you're not going to get neural tube development. And so that part of organogenesis is not going to complete. You're not going to get a central nervous system out of this. So that will completely disorganize embryological development. There's no way that it can complete normally under those circumstances. So I mentioned early on in the presentation that two very key steps that we need to talk about as we consider the progress from a single cell, the zygote, to a multicellular organism with many different types of cells organized into different types of tissues and organs. We need to be able to explain how those cells know what structure they're in within the three-dimensional body of the organism and what specific types of cells they're going to become within that particular structure. And the two processes that explain that are referred to as determination, which basically tells the cells where they are in 3 dimensions in the body of the organism and therefore what structure they're participating in. And then differentiation, which tells them specifically what type of cell they're going to become because of their position within that structure. And why this is important is because every different type of cell, and there are 220 different types of cells in your body, for instance, is that type of cell because it uses only a subset of the total number of genes in the genome. And in order for a cell to become part of a structure and the particular types of cells within that structure is a function of differential gene expression, switching on the necessary genes to become that type of cell and making sure all the other genes that are unnecessary get switched off. And they can only do that by having information about where they lie within the body of the organism. So there has to be some way of signaling to the cells where they are and what they need to become, determination and differentiation. And this is wrapped up in an idea called cell fate. So how does embryological development trigger cell fate? That's basically the question that we're asking here. So there are two basic processes that can be involved here. So somewhere along the way during the early development of the organism, the embryonic cells have to take on different characteristics from each other. They can no longer be identical. They have to take on some differences. In other words, the cell mass has to become heterogeneous, different types of cells that will follow different developmental pathways and wind up as different types of cells. Now, for some organisms, that starts on the basis of the determinants that are placed in the egg cell by the mother, these so-called cytoplasmic determinants. So you can see here the presence of determinants in a fertilized egg of a little worm called C. elegans. And the reason why C. elegans is a really good model organism is because it can be seen under a microscope. It is a tiny worm. It's only about 1 to 1.5 millimeters in length. So it's just on the limit of resolution with your naked eye. But they are easily visible under the microscope. They're easy to grow in the lab. They're quite simple. And as has been discovered, their body contains exactly 959 cells. And as a consequence of some very, very clever and very, very determined research, the origin of each one of those cells, each one of those 959 cells, is known. We can trace the origin of each one of those cells back to the zygote. We know precisely how they were arrived at. And one of the reasons why those 959 cells emerge as different types of cells is because in the egg cell, the female places these cellular determinants, and you can see them highlighted here in a fluorescence microscope. These glowing spots are the determinants. So this is what you see in the newly fertilized egg. They have a symmetrical distribution. But after the first cleavage takes place, and we have the first two blastomeres, one blastomere completely lacks the determinants, and the other blastomere has all the determinants, and they are located at one end of that cell. So how did we get that? Well, here you can see the zygote undergoing its first division. You can see that it's in probably metaphase. There are the chromosomes sitting in the middle of the metaphase plate. And you can see when we look at it under fluorescence that all the determinants have migrated to one end of the zygote even before the cell has divided, which has led to all the determinants showing up in this blastomere. When these two cells divide, all of the determinants are located in this cell. It is distinctly different from the other three cells because of the presence of the determinants. And so simply because of the distribution of these determinants during these first rounds of cleavage, we've created a polarity. Determinants at this end, no determinants at this end. We have a polarity in the embryo, and that immediately establishes, in this case, the anterior posterior axis in the C. elegans worm. Once you have those asymmetries, then there needs to be some sort of signaling, some interaction between cells or between some regulatory mechanism that starts the cells down the pathway to their final fate. Again, this would be changes in gene expression. So we can track the movement of cells down these developmental pathways in a number of different studies. So the early studies were done by a German developmental biologist by the name of Hans Spemann. And Spemann is probably the person who really deserves the credit for establishing developmental biology as a discipline, because he did a lot of the early work in establishing the role of different tissues in determining cell fate. So let's take a look at some of his early experiments that allow us to map out the fate of individual cells and groups of cells. So here is some of Spemann's early work replicated in another organism. So this is what we have shown previously in terms of color coding the tissue during early gastrulation, and where that tissue winds up early on in organogenesis. And you can see mesoderm located here in the early gastrula, forming the mesoderm layers here. Endoderm in the early gastrula, endoderm in early organogenesis, ectoderm in early gastrulation, ectoderm in in early organogenesis. This is based on some of this early work done by Spearman. So how do we, in modern terms, carry out a similar sort of fate mapping? Well, we have available to us now non-lethal dyes, non-toxic dyes that you can inject into any cell within a developing embryo. So in this case, it's a 64-cell embryo. This was probably a little bit past the morula stage. It probably has a blastocele, a developing blastocele on the inside. In this particular situation, the dye has been injected into this cell only. And all the daughter cells of this cell receive a portion of the dye because it's incorporated into the cytoplasm. And here are all the daughter cells in the larval stage of an organism called a tunicate. This is actually, a tunicate is a chordate. It is, however, an invertebrate chordate, so it belongs to one of the two invertebrate clades of our phylum, phylum Chordata. Remember, we belong to the subphylum Vertebrata. And it turns out that the tunicates are the chordate invertebrate clade most closely related to our own. So this is somewhat interesting in that sense. Also, tunicates undergo a huge change in body plan from the larval stage to the adult stage, but more of that in chapter 34. So here are the daughter cells of this cell in this early blastular stage. And you can see that they are the daughter cells because they contain all the dye. If we inject this cell, which is right there on this one, this cell is right there on this one. If we inject the dye into this cell, these are the daughter cells of that cell in the larval stage. So we have the ability to track individual cells now. Spearman couldn't do that in his early work. He had to track groups of cells, and he didn't have all of these nice non-toxic dyes that we could play with today. He had to work with other chemicals and other ways of dealing with it, but he was still able to map out in a block fashion where the tissues would wind up in the embryos. So here is a similar map for C. elegans. So you can see down the bottom here a picture of an actual photograph of an individual worm, tiny little worm. Here are all the body organ systems that we can identify. And here is the origin of all of those individual cells that are produced. And here's the other reason why C. elegans is a really popular model organism, because you get this completed worm in 10 hours post-fertilization. So you don't have to wait very long to see everything. So here it's only laying out tissue by tissue the origin. But remember, we can actually lay out now in the map the origin of every one of the 959 cells that every C. elegans possesses. And this is in fact the case. Every individual C. elegans contains exactly the same number of cells, 959 cells. It's quite remarkable, but it makes it a very, very useful model organism for a number of reasons, not just in understanding embryological development. There are other things that we can do with it as well. So how do we go about establishing cellular asymmetries? Well, it depends on what type of organism you're looking at. We've seen in C. elegans, which is an invertebrate, how that could be established. And this seems to be a fairly consistent pattern in most invertebrates. But what about vertebrates? How do we establish axes in invertebrates? Well, we've already seen in frogs a way in which that can be done. So here is, again, something that has been demonstrated by Spearman's lab and some of his early work. So again, we've talked about what happens when the sperm enters into the egg cell. We already have the anterior posterior axis established by the orientation of the animal hemisphere and the vegetal hemisphere. Remember, the actual anterior posterior axis is not pole to pole, but it is hemisphere to hemisphere. When the sperm enters in, you have the melanin containing cortical layer rolling backwards to expose the unstained region and gives you the gray crescent, and that gives you the dorsal ventral axis. And remember, once you get two axes established, you fix the other axis. So the left-right axis has been established here. If this is dorsal and ventral, anterior and posterior, this would be left, and the other side would be right. Then you get the first plane of cleavage, bisecting the gray crescent in this case, splitting it from animal to vegetal. Well, what Spearman did was manipulate this. So again, we have a egg cell that has been fertilized. We've got the gray crescent. We get the first cleavage plane, again, vertical, animal to vegetal. What Spearman then did was tease these two blastomeres apart. And because of the nature of these two blastomeres, they contain all the necessary information to maintain the three body axes. These two cells would then develop into two normal frog embryos, tadpoles. And they would be identical to each other. In fact, this is the basis for what we now know is the origin of identical twins in humans, for instance. Now, it's not the first two blastomeres that split. It's usually during that period when you have a small ball of cells. So it would likely be probably in the morula stage, probably in the 8 to 16 cell stage. Something happens to split that cell mass into two. And because those cells are considered to be totipotent, it's a term that I will mention again a little bit later, but the term totipotent simply means that they have the capacity to become every particular cell type present in the body of the organism. And so when a human embryo splits, a cell mass splits into two, those blastomeres in those two resulting cell masses, if this happens early enough, remain totipotent and develop into two embryonic cell masses, two trophoblasts, that then implant in the wall of the uterus and develop into two individuals that, because they are the product of a single egg and a single sperm, are genetically identical twins. Conjoined twins, what are sometimes referred to as Siamese twins, occur when the split is incomplete and there is a shared group of cells, and you get two individuals develop that share a common group of cells. And where that common group of cells forms when this incomplete splitting occurs determine what body features are shared between the two conjoined twins. Just to finish off the story, if you're talking about non-identical twins, what are sometimes referred to as fraternal twins, well, they are the product of two separate eggs being fertilized by two separate sperm. They are genetically non-identical, but you get two eggs released that are both fertilized, and they develop into two individuals that are considered to be twins because they shared the same uterus, but they are non-identical because they are the product of two different eggs and two different sperm. So what did Spearman do with this? Well, in his experiment, he again allowed the egg to be fertilized, but this time before the first plane of cleavage developed. He took a thread and wrapped it around the zygote in such a way that he could very carefully create the first cleavage, but create two blastomeres that were no longer equivalent. One blastomere contained the gray crescent, the other one lacked the gray crescent. So this particular blastomere and the daughter cells that would develop from this blastomere, because it still had the ability to divide, lacked information for the dorsal ventral axis. And so as a consequence, they developed by the default programming and developed into ventral features only, the belly. Meanwhile, the other blastomere containing the gray crescent contained the necessary information to establish the dorsal ventral axis, because it's got the gray crescent, which remember establishes dorsal. And so you have the resulting blastomeres, daughter cells, establishing a developing embryo that has the dorsal ventral axis information, and so it develops into a normal tadpole. And this clearly confirmed the role of the gray crescent in establishing the dorsal ventral axis. So in the embryonic development, we start out with cells that are totipotent. They then become restricted in terms of their potency. And they are, I'm having a bad day. I honestly cannot recall all of the terminology that I want to pass on to you. Maybe I shouldn't, because we're digging very deep into some ideas. But we start out with cells that are totipotent. The potency is restricted until final fate is determined. So the totipotent cells in humans are often referred to as fetal stem cells. So when you hear the term fetal stem cells, what's really being referred to are these cells that have this capacity to become any of the cell types that are found in the human body, all 220 types. As development begins and cells start following their developmental pathways, their ability to become different types of cells is progressively restricted as they move further out on the branching of cell types. Ultimately, they reach their final twig when they are locked into what they are ultimately going to become. And we say that they have been determined at that point. So how is this determination developed? What is the signaling that's going on, or what's the origin of the signaling that goes on to determine a cell's fate? Well, again, we turn to the work of Speeman and one of his graduate students, Hermione Mangold. Unfortunately, Mangold does not get as much credit as she should for the work that she did, and in part that's the fault of Speeman, because Mangold unfortunately passed away very young, and Speeman actually took substantial credit for a lot of the work that was actually Mangold's work for her doctoral dissertation. And so A lot of the time you see this referred to as Spearman's work, but in fact, it's Spearman and Mangold. And Mangold was the graduate student who actually did the vast majority of the work that we're about to talk about. So one of the things that Spearman and Mangold realized once it had been established that the Grey Crescent established dorsal ventral was that the blastopore develops in that region where the Grey Crescent is. And so they started to become curious about what it was in that region that might be causing the fixation of the dorsal ventral axis. It had to be associated with something on the dorsal side of the embryo. But given that the dorsal ventral axis established an axis that created a region 180 degrees opposite to the dorsal side that establishes that became the ventral side, it could just be that perhaps something on the ventral side was establishing the dorsal ventral axis. They needed to determine what was going on. So they took advantage of the fact that salamander embryos come in two forms, a highly pigmented form in some species and an unpigmented form in other species. And they transferred tissue from various locations in the pigmented embryos into different locations in the unpigmented embryos. And the one piece of tissue that turned out to be significant was what is referred to as the dorsal lip, the overhanging ectodermic tissue adjacent to the forming blastopore, what is known as the dorsal lip of the blastopore. When that is transplanted from a pigmented embryo into an unpigmented embryo and located 180 degrees opposite the existing dorsal lip, what was generated was an embryo that had a second embryo that aligned belly to belly with the non-pigmented embryo. Now, what was most remarkable about this, other than the fact that you get two complete embryos, was that the secondary embryo contained mostly non-pigmented tissue. In other words, the vast majority of tissue developing in the secondary embryo was created from tissue in the primary embryo, unpigmented tissue, not from pigmented tissue as you might expect. So why was this the case? Well, it turns out that the dorsal lip belongs to a family of tissue known as inducers. This was, in fact, the first inducer to be recognized. It sends out chemical signals that establish the dorsal versus ventral side of the embryo. And it's actually quite a curious arrangement that goes on here that establishes dorsal ventral. There is a protein that is now known to be involved in this. It's called BMP4, bone morphogenic protein. It's actually a growth factor that is involved in inhibiting dorsal growth. And when you inhibit dorsal growth, what you get is ventral. So BMP4 is generated on the ventral side of the embryo, and its presence inhibits normal dorsal growth. And so you get ventral. But if you introduce a dorsal lip and place it where the ventral is supposed to go, now you get a strong dorsal signal and you get a very weak ventral signal. In fact, in this region, you get no ventral signal at all. And so you get dorsal development. And in the intervening region where BMP4 now gets expressed, you get ventral. So this is an example of an inducer, the dorsal lip of the blastopore, overriding the BMP4 signal in its location and inducing the development of dorsal. In the absence of the dorsal lip, the BMP4 triggers the formation of ventral by shutting down the signal for dorsal. It's an interesting way of controlling the development of the tissue, but this is the way it works out. And this is something that has been more recently recognized in the last 20 years or so. And we're still working on understanding inducers. We now know that there are other inducers involved. The dorsal lip of the blastopore is not the only inducer that we know of. There are other inducers involved. In fact, we're going to take a look at another inducer that is involved in the regulation of vertebrate limb formation, because this is also an interesting one. And there's a lot more known about this one because the manipulation has been done in chick eggs. It's also been done in Drosophila, and we know that the proteins that are involved here are extremely highly conserved. So I've mentioned previously the role of Hox genes. These are master control genes that regulate families of other genes. And these establish body parts establish body plans. Or patterns. So remember, what we're trying to establish here is why, once you've got the three axes in place, why certain structures form in certain locations with respect to those axes. So we've gone over some mechanisms that establish the axis. We've seen determinants can do that. We've seen that random acts like sperm penetration into an egg cell can do that. We've seen that inducers can establish body axes. What we're going to look at now is how cells can be told what position they occupy with respect to those axes, because that establishes body plan or body patterns. And so the vertebrate limb, and particularly the chicken limb, has been studied quite a lot. The wing itself has been the subject of a lot of study. And the reason why is because it's very easy to manipulate chick embryos inside the egg. You can cut the window into the shell and tease aside the interior membrane, the chorion, without disrupting the development of the embryo. And so long as you do this under sterile conditions, the embryo can still undergo its full developmental pathway. And you can then manipulate using micro manipulators and microscopes the chick embryo and insert things, move things around within the embryo. And because the embryo develops fairly quickly, you can see the effect of those changes pretty quickly. So, as I said, the genes that are involved in the vertebrate limb have been found across the board in the animal kingdom. So they're found in Drosophila, they're found in chickens, they're found in humans and vertebrates in general. So these genes are very important. In the fruit fly, the gene is known as hedgehog because it not only is involved in establishing limbs, but it also is involved, the legs, part of it, but it's also involved in establishing the little hairs that are found on the surface of a fruit fly. In chickens, it's involved in the forelimb, and also in humans, it's involved in the forelimb. But it's not called hedgehog, as you'll see. So when we take a look at limbs in vertebrates, they are produced by what are called limb buds. And we always have 4 of these if you're a terrestrial tetrapod. 2 for the forelimbs and 2 for the hind limbs. And they have essentially the same basic structure. The bud has a ridge of tissue, which on the forelimb is referred to as the apical ectodermal ridge. And it is an organizer that establishes one of the 3 axes of the limb. So let me see if I can show you what the 3 axes look like. So I'll do it on my hand, but remember this is established on the limb itself. So it does have a anterior posterior. So the thumb is considered to be anterior and the pinky is posterior. So there's your anterior posterior axis. Dorsal ventral is back of the hand dorsal. Palm of the hand is ventral. And then for the 3rd axis, rather than left and right, because a limb doesn't have left and right, we have what is called proximal and distal. Proximal is close, so that's the shoulder, and distal is far, so that's the fingertips. So those are 3 axes in a limb, and all limbs have those basic axes, but we only need to really establish 2 of them. So the proximal distal axis is established by the apical ectodermal ridge, which has a gene that secretes a protein that establishes a gradient from the AER at the distal end yeah, I'm not sure why it keeps doing this. At this point, I want to keep those because let me go back and share this again with you. So we're back again. So let me just continue with this and put the line in. So that's the axis that you're going to see. And that establishes the proximal distal. Remember, proximal is at this end. So this is proximal, this is distal. And then the anterior posterior axis is established by another region called the ZPA, the zone of polarizing activity. And it's located in what will eventually become the armpit in humans. And it also secretes a protein that creates a gradient going this way. So again, we have the anterior at this end and the posterior at this end. So there's your gradient. So we've established 2 of the axes. We've got the proximal distal, thanks to the apical ectodermal ridge, and we've got the anterior posterior, thanks to the ZPA. And both of these produce a gene product, a protein, that establishes these axes. And both of these genes are actually members of the Hox gene family. So ZPA produces the protein that is related to Drosophila's hedgehog. So when the study of the chick limb bud began, it was suspected that something like hedgehog might be involved because of hedgehog's role in the organization of the Drosophila. And so once hedgehog was identified in Drosophila, they went searching for a similar gene in chickens and discovered the gene in chickens. And it was then found that the ZPA was the source of the gene product. And so that established the ZPA as the source of this gene, and therefore potentially establishing the anterior-posterior axis. But how to establish that that was in fact the case? Well, the way to establish that, and this is just explaining the axes as I showed you on the camera, so we'll move past this. The way to establish that in fact the ZPA is the origin of the anterior-posterior axis because it's producing this protein is to take a donor ZPA and transplant it to the anterior side of an existing limb bud, just as Spearman and Mangold did with the dorsal lip of the blastopore, to establish that the dorsal lip was the inducer or the organizer of the dorsal ventral axis in the frog. So the researchers who did this work transferred the ZPA to the anterior side of an existing limb bud and took a look to see what happened. And this photograph is an X-ray of the resulting chicken limb. And just so that you can orientate yourself to this, what you've got is a chicken limb that is like this. I don't know where, yeah, let's see if we can get it. So it's arranged like this. And so you've got two dorsal sides, which remember, two posterior sides. Remember, posterior is the little finger, the pinky on our hand. So that's it there. And so this is the pinky equivalent, number 4. Maybe I should write that in here. Let's try that again. Pinky side. And this would be, and this again would be pinky side. So you've got these, a similar sort of product that Spearman and Mangold saw with their salamander embryos, where you have two dorsal, or in this case, two anterior sides, so two posterior sides with a merged anterior side in the center. Now, that simply established that, yes, the ZPA is establishing anterior-posterior axis. But was it, in fact, the product of the ZPA that was equivalent to hedgehog in Drosophila that was doing this, as suspected? The way this was figured out was to actually clone the gene and create the gene product, the protein, and then take a polystyrene bead and soak it in the protein and transplant the polystyrene bead into this location. And the end result was an identical result. So I'm going to put in here, replacing the ZPA with a bead soaked in hedgehog protein. The same result. So yeah, I'll put it in quotes here because it wasn't actually hedgehog, it was the equivalent of hedgehog that was used. But in subsequent experimentation, they actually took the hedgehog gene and used that and got the same result. And this is one of the things that you notice about the Hox genes and their gene products. They're incredibly highly conserved across evolutionary time, and you can basically take the equivalent gene from one organism and transfer it into a distantly related organism without any change in function, at least with our experimental models. Now, where it gets interesting is that once it was recognized that this was the case, the question was then asked, in humans, is there a hedgehog equivalent to the chicken protein? So again, and this was prior to the Human Genome Project, so it was a little bit more difficult, but they went searching for an equivalent gene and found one. And it has an interesting name. And I suspect it got the name because it was a graduate student who named it. It is called Sonic Hedgehog, after the game character. So we now know that, in fact, our limb, and particularly our arm, is the product of the same type of development. There is the equivalent of a ZPA in our limb bud. It establishes anterior-posterior. There is an apical ectodermal ridge that establishes the proximal distal axis, and our limb develops along the gradient of these proteins, because that's in fact what the cells are recognizing. They detect the levels of each of the proteins. That provides them with three-dimensional information about where they are in the developing limb, and that in turn, with extra signaling, tells them what type of cells to become on the basis of their three-dimensional location. So they know that they're in a limb, and then they know what part of the limb they're in, and that finally determines the type of cells that they will become. So it is the Hox genes that are the genes that are behind the establishment of the pattern. And when we look at the animal kingdom, what we recognize is that it's the number of Hox genes that determines the complexity of the animal that you wind up with. So the way we now look at the animal genome is to say that there is a core set of genes for animals. And if you want an analogy to think about baked goods, pastries, biscuits, cakes, those sorts of things. There is a basic recipe for all of those using the same set of ingredients, just as there is a basic genetic recipe for an animal using the basic set of gene ingredients. What determines the final product when you're making baked goods like bread and pastry and the like is the ratio of the ingredients that you mix together, when you mix them together and how you mix them together. The way that you cook it, whether you bake it, boil it, fry it, whatever, the conditions under which you do it and the temperature under which you do it and for how long you do it. And once you take into account all of those variables, you wind up with a particular type of baked good based on what you did. The same is now true about the way we think about an animal. There's this basic core of genetic ingredients that make up an animal. What determines the final form of the animal that you're going to get is the way you utilize those gene products, and that's under control of the Hox genes. And so for invertebrates, when we look at the genetic origin of the animal kingdom, invertebrates have a single set of Hox genes, a cassette of about 7 to 9 genes. When we transition from invertebrates to vertebrates, we see a doubling of the Hox genes, which means we have a greater number of Hox genes, which means we have a greater variety of genes to control the basic recipe. In other words, we can start to use the basic ingredients in more ways and more novel ways, which leads to the origin of vertebrates. But early vertebrates lacked jaws. So we will encounter jawless vertebrates as the earliest vertebrates. When we transition from jawless vertebrates to jawed vertebrates, we see another doubling down on the Hox genes. We go from 2 complete sets to 4 complete sets, which now creates the opportunity to do substantially more with this basic core set of genetic ingredients. And so we see, as we go from invertebrates to jawless vertebrates to jawed vertebrates, an increasing level of complexity. Not because there are more genes involved, but because there's more room to utilize the ingredients in new ways and in more novel ways to create new levels of complexity that were not possible with a limited number of Hox genes. It's the best way to explain why a fruit fly is the product of 17,000 genes and a human is the product of no more than 23,000 genes. It has to do with not the physical number of genes that are involved, but the way you utilize those. How many Hox genes do you have to create multiple layers of control over the utilization of those basic genes? And just to illustrate that, remember again that the human equivalent hedgehog, sonic hedgehog, is known to play a role in the development of human limbs. Well, it turns out that sonic hedgehog also is involved in the development of the brain, but it doesn't create a limb where the brain is. But that's because hedgehog is operating in a different chemical environment, because it's operating in an environment with a different combination of Hox genes than the combination of Hox genes that are seen in the limb. And so now you have different structures. Remember, different ways of utilizing the ingredients in different conditions results in different types of biscuits and cookies and so forth. So when you start using the genetic ingredients in combination with a different set of Hox genes, you wind up with different products. And that's why you can create the very complex 10 trillion cell with 220 different cell types human body from such a small number of genes. It's totally rewritten the way we have to think about the genome, whether it be a human genome or the genome of any other organism. We will address some more of this as we talk about the animal kingdom, because it really does give us a window into the origin of the animal kingdom and its evolution. But we'll touch more on that. But we need to move on. So this is a photograph of a young man or a child. I don't know whether it's a young man, but I've seen video of a young man who has this condition called polydactyly. It occurs about 1 in every 400 births amongst humans. And it's not unusual for surgeons to remove the extra digit early in the life of the individual, but it's also not unusual these days either for the digit to be maintained. Now, when polydactyly occurs, the extra digit is always on the pinky side. So there's the extra digit. Notice it's on the little toe side or the pinky side. And the reason why is, again, because the anterior to posterior axis in the foot is the same as the anterior posterior axis in the hand. And remember, that runs from the thumb anterior to the pinky posterior. So this is the posterior to anterior. So this is post, and this is ant. And sonic hedgehog is expressed here. And it turns out that in polydactyly, it's overexpressed. So one of the interesting things that we've now started to realize is that the level of gene expression determines the number of digits. And one of the things that you're going to discover is that in the early record of terrestrial tetrapods, a limb did not have 5 digits. It could have had up to 10 digits in some of the very, very earliest ancestors of amphibians. And almost certainly, this is a product of an overexpression of the hedgehog equivalent in amphibians, or at least that's the hypothesis. So we know that polydactyly arises because of a mutation that leads to an overexpression of sonic hedgehog. That also explains why the normal condition is to have 5 digits. Apparently, the expression of sonic hedgehog has changed over time, and the quote unquote normal level of expression provides 5 digits, and the decreasing level of expression leads to changes in the digits from the area of expression, the pinky side, to the area of lowest expression, the thumb side or the big toe side. So a lot of what we observe, we are now starting to be able to explain genetically, but there's a lot more to do here. There's a long way to go before we fully understand this, but that gives us some more ideas. So the final piece of the puzzle in laying out body plan is to talk about the role of cilia, which you may think, well, that's kind of strange here, considering that we're talking about the role of genes. Well, it turns out that there are 2 different bodies of cilia produced in human bodies, for instance. And some are motile, as we would expect, and those would be the sort of cilia that we see lining the bronchial passages and attached to the end of a human sperm, that provide movement of fluids over the cell, which in the case of the sperm means that the sperm moves forwards, or in the case of the bronchial passages, moves material out of the lungs and sweeps dirt and other things that may have been inhaled up into our throat so that we can either swallow it, which is mostly what we do, or spit it, which we do sometimes if it gets particularly nasty. But there are also populations of cilia that are non-motile, and this is the product of a mutation. And those non-motile cilia, for instance, are important for the normal development of the kidney. But it turns out that there is a condition called Cartagener's syndrome that is a product of motile sperm possessing a no longer functioning correctly, let's put it that way, and that those motile cilia actually now become non-motile or monocilia, and that has an effect on the arrangement of the organs in the body cavity of humans. So anyone who has Cartagener's syndrome suffers from a situation, well, not really suffers, but has a condition called situs inversus, meaning that the orientation of the body organs in the body cavity are rotated through 180 degrees from their normal orientation. And so, for instance, the heart is offset to the right rather than being offset to the left, as we would expect. Notice that the liver is normally on the left side. In situs inversus, it's on the right side. Everything is flipped around through 180 degrees. And the reason why this, we think, is occurring is because motile cilia during embryological development set up a fluid rotation in the body cavity that results in the fluid moving from the right side to the left side. So that would be the normal flow. So when we view it here, it's clockwise. In situs inversus, the cilia are non-motile, and so there is no fluid flow. And that seems to be the cause of situs inversus. In the absence of this leftwards movement of the fluid in response to the motile cilia, the organs are rotated through 180 degrees. In terms of normal organ function, there's no difference. The individuals with situs inversus do not notice any difference in normal organ function. And typically it's picked up fairly early when a doctor uses a stethoscope to try and hear the heartbeat. The normal routine is to put it on the left-hand side of the body cavity because that's where the heart is supposed to be located. Don't hear it, move it to the right-hand side and hear the heartbeat and realize that the individual has situs inversus. There are other features that are associated with cardiagenous as well, but this is one of the more curious ones as far as we're concerned in terms of body plan and patterns. So that gives you some background to the way embryos develop and why it's important to us as evolutionary biologists to understand the relationship between evolutionary origin and relatedness and development of embryos. As I said at the beginning, when we follow embryological development, what we can do as a way of getting clues to evolutionary origin and evolutionary relationship is to track the developmental pathway of the embryo and look for points of divergence. When there is a greater degree of commonality in embryological development, that is usually an indication of greater evolutionary relatedness. When the divergence occurs a lot earlier on, that is usually an indication of more distant evolutionary relatedness. And we'll see that played out as we continue our investigation of the animal kingdom in the remaining 3 chapters for this semester. In the meantime, let's look at this last concept check question and then close out the recording. So, how do axis formation and pattern formation differ? Remember, pattern formation is the location of different limbs or different body systems in 3 dimensions in the body. Where does that pattern formation come from, or where does the information for that pattern formation come from? From the axis. So the axis formation is a product of the asymmetry that starts to develop in the developing embryo that leads to polarity in the embryo. That leads in turn to the establishment of the 3 axes: anterior, posterior, dorsal, ventral, and left and right. Location with respect to those axes is what establishes the pattern. That's why the limbs are in certain locations. That's why your arm, for instance, has the particular pattern that it has with the distribution of tissues and the distribution of different features within the arm, because the cells are responding to their 3-dimensional location with respect to the 3 axes of the limb. So the axis formation is derived typically through Hox gene information and is a product of the asymmetries that develop in the embryo that creates the polarity that leads to the 3 axes. And then pattern formation plays off those 3 axes by using position with respect to the 3 axes to establish what features should be in each location within the body of the animal. So that's the difference between axis formation and pattern formation. So that's it for this chapter. We will return again next time and take a look at the basics of the animal kingdom in a very short chapter that looks at the basic origin of the animal kingdom, big picture time. And there we'll look at some changes that have occurred over time in the way we understand the animal kingdom as a consequence of genetic information and a more detailed examination of evolutionary relatedness as a consequence of this evo-devo work and genetic sequence analysis. So until then, if you have any questions, concerns, or comments about anything that I've covered here, by all means, come and see me during office hours. Otherwise, I'll see you back here again for our continued look at the animal kingdom. Bye for now.